Hi there, thank you for your interest in our project. My name is Jialin Liu, and I'm a PhD candidate from the University of Michigan. I'm going to give you a brief walkthrough of our project called Integrative Single-Cell Genomic Analysis Identifies a New Type of Skeletal Stem Cell in Bone Marrow Endosteum. So first, a little bit of the background. Skeletal stem cells provide an important source of bone-forming osteoblasts and support overall bone health. The identity of SSCs in aged mice are already identified, but the identity of the SSCs in young mice are still unclear. So in order to find candidates of a putative stem cell cluster, we first performed 2 rounds of single-cell sequencing, separately from a young mice aged at P21 and an old mice aged at P18 months, and we integrate them with our previous published integrative analysis tool, which is also called LIGER. And here, strikingly, we found this really interesting cluster called osteostem cluster, surrounded by the osteochondrocyte, the osteoislands, the chondrocyte islands, and the adipocyte islands. And to further validate this putative stem cell cluster, we infer the differentiation and the latent time for each of the cells using using SC-value and cell ranks. So here on the left-hand side, the SC-value results shows these OCT stem cell cluster kind of serves as a cell of origin cluster. You could see these arrows are coming from all of these cluster pointing to the other clusters like osteoblasts and reticular cells, and also the results from the cell rank suggest a similar story. You could see these OCT stem cluster has a really high possibility of being initial cell state. And also on the density plot, we could see these specific cluster is enriched in the young bone but depleted in the old bone. And to further validate to further confirm the potential of the stem cell cluster, we perform another round of sequencing, which is we do a single cell RNA-seq and a single nucleus ATAC-seq on the young bone marrow. And we developed this novel way of demonstrating the differentiation potential towards each of the 3 terminal states. So here we define the reticular cells, osteoblasts, and chondrocytes have the 3 terminal states. So let me explain a little bit more about these simplex plots. So in these plots, each dot on the plane represents a cell, and each arrow represents how the RNA velocity is going towards each of the 3 terminal points. And also each vertex represents the proximity between each of the cell to the 3 terminal states. And overall, we think the simplex RNA plots tells us the OCT stem has a affinity with the osteoblasts and reticular cells. However, the chromatin accessibility, which is measured by the single nucleus ATAC, suggests the OCT stem cluster has a similarity between the reticular cells and the chondrocyte cells. And next, we also do we use Homer to do some TF bonding motif. And from this analysis, the OCT stem cell is similar to the chondrocyte. Finally, we proposed this really interesting model, these OCT stem cells with osteoblast chondrocyte transitional identities have reticular-like chromatin accessibility, and it has further differentiation potential towards adipocyte cell and osteoblasts, and the enrichment of TF bondings and has the enrichments of the TF that regulate chondrocyte differentiation. And we also did some in vivo validations. We constructed these really interesting transgenic mouse model, which allows us to showing the FGFR3 activation over time. And then we performed a drill hole surgery on the bone marrow, and and after 7 days, we could see the injury area was filled with the FGFR3 marked cells, and however, as a control, the GAS1 marked cells are already depleted. So overall, these evidence are suggesting the FGFR3 marked endosteum putative stem cell are dictate active osteogenesis in injury. And next, we ask what will be the potential role of these FGFR3 plus endosteal stem cells in pathological conditions. So a little bit of the background again. The osteosarcoma is a bone tumor with an unknown cellular origin. in which P53 encoded by TRP53 is identified as a driver gene in humans and mice. So here we build another like transgenic mouse model to allow for like a joint control of FGFR3 and TRP53 gene. So you can see here, like strikingly, after 9 months, like 5 out of 17 mice developed these huge tumors radiating from the bone marrow, which destroy the pre-existing cortical bones. And next, we definitely want to understand like what molecular features represent these tumor cells. So again, we did another round of sequencing. We do single cell sequencing from the control young bones, and we also do single cell sequencing from the young bones with tumor. And again, we integrate them with Liger, and to identify the tumor cells from the single cell RNA sequencing data sets, we used some published software, public tools for copy number variation analysis, such as the copycat and the infer-CNV. And you could see like the putative tumor cells include the whole chondrocyte islands, and also the OC stem cells stays between the main island and the chondrocyte island. And we then dive a little bit deeper and perform further experiments. So we first replotted the results from the copycat, and you could see like there are significant copy number changes across the entire genome, including both gain, which is colored in red, and loss, which is colored in blue. And we also perform the whole exome sequencing and it's basically suggesting the same story. We're seeing like the genes with significant loss, which is colored in blue again, and also seeing regions with significant gain, which is colored in red again. And finally, we do a gene oncology analysis using the differentially expressed genes in the tumor cells. And it suggests some really interesting terms related to tumor development, such as like the positive regulation of proliferation and the response to external stimulus. So as a takeaway message, integrative analysis revealed a novel population of bone marrow stromal cells with osteoblast chondrocyte transitional identities, and also OCT stem cells coordinate active osteogenesis in young mice. They are abundant in young mice, but are depleted in the old mice. Finally, OCT stem cells contribute to aggressive osteosarcoma-like lesions upon the loss of the P53 tumor suppressions. And these derived tumor cells show obvious abnormal molecular features. And if you would like to know a little bit more details, please feel free to refer to our recently published article in Nature Communications in April 2023. And that will be the end of my presentation, and thank you very much.